The eLife Podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.
Welcome to this very special edition of the eLife Podcast. Why is it special? Because it's our 50th episode, and we've decided to mark that milestone by focusing on a field that's huge and tiny both at the same time. Huge in terms of the rate at which the discipline is growing and the impact that it's set to have on our lives, and tiny because its subjects are microscopic. If you've not guessed it yet, it is of course our microbiome, the community of microorganisms that live on us and in us and outnumber our human cells by maybe 50-fold. We're literally passengers in our own bodies. Hello, I'm Chris Smith from The Naked Scientist, and in the next 30 minutes we're going to learn how gut bacteria might alter your risk of diabetes and how the microbiome can even manipulate your mood. But first, beginning at the top end and with what we put into our mouths, Sarah Durienzi has been looking at the effects of feeding mice on a diet enriched with linoleic acid, an essential fatty acid that we eat an awful lot of these days.
So I got to thinking about what we do in our daily life, and that's that we eat a lot of food, but what we eat from day to day frequently changes quite a bit. And that food that we consume, this is exactly what our microbes get to feed off of. So if we're changing every day what they get to see, you would think that we would be changing what microbes live in our intestine every day. But that's not true. Over time, we actually see that our gut microbiome is reasonably stable. And so a big broader question is, how do we maintain such a stable gut microbiome? Either the gut microbes themselves have mechanisms in order to deal with a rapidly changing diet, or there's the host gut environment is somehow buffering that direct interface between the diet and the gut microbe and changing how that interaction occurs.
Indeed, microbiologists often reassure people, even lonely people, that thanks to your microbiome, you never dine alone. But the point is that this is really hard to test, isn't it? Because the microbiome is very diverse, diets are very diverse, people are very diverse. So how have you sought to control this and try and turn those enormous numbers of variables into something tractable?
Well, I also just want to bring up that this question is actually even more complicated. When you're really talking about the diet that we eat, this is really a question for the small intestine and not a question of the large intestine. It's less than 10%, sometimes even only 1% ever really reaches the large intestine.
Okay, so just talk us through the experiment then, and essentially what you did.
So if we follow with the very simple hypothesis that when we consume dietary fat, it's broken down into its individual fatty acids, and those individual fatty acids are what interact with gut microbes. So what we can do is to simply take those individual fatty acids and directly put them on gut microbes that are found in the small intestine. These fatty acids, so for instance like linoleic acid, directly inhibits gut microbes that are found in the small intestine.
That's rather strange, isn't it? Because linoleic acid is one of the essential fatty acids. We can't make that in the body through our own metabolism. We're absolutely dependent on our diet as a source of that. So it seems rather counterintuitive that something we're obliged to eat is going to poison our microbiome.
Exactly. Moreover, linoleic acid is also the fatty acid that we consume the most of. It's highly abundant in vegetables and nuts, so it's found in all of your vegetable oils. These are foods you eat every single day.
And that being the case, then if it's that toxic, we shouldn't have any of these microbes left in our microbiome, should we?
Yeah,
We should have driven them all should have driven them all out of existence.
Exactly.
So something's going on.
Exactly. So then we get to, let's see what actually occurs in the small intestine. So to do this, what we set up was a long-term diet study. In this study, we designed diets that were very carefully controlled and just differed in their percentage of fat versus carbohydrate. And the specific fat that we used in this diet was soybean oil, rich in this linoleic acid. So by putting mice on these different diets, we can see what happens to their gut microbiome. So when we do that, really kind of surprisingly is you don't actually see very big changes at all, which is not what you would expect based off of what we see that happens in a test tube.
One of the perils of extrapolating to the in vivo situation, what goes on in vitro, isn't it, this?
Right.
It suggests then that something must be endowing the microbiome when it's in vivo with more resilience. Either there's some protecting factor or something else is going on.
Yeah. So we also took this study a step further. We used a compound called propynium monoazide. And what it does is that it allows us to identify microbes that have been affected by the inhibitory effects of linoleic acid. So what we see is that those gut microbes that were inhibited in the test tube, they're not showing that inhibition in the gut. In fact, they're actually doing better than some other gut microbes. So in fact, what we're seeing is that some of these gut microbes are not directly seeing linoleic acid in the gut the way that they directly see them in a test tube.
So that would suggest then that something is getting in the way of the access of that chemical into those microbes.
Correct.
Or that the microbes themselves are doing something different when they're in situ in the gut where they're supposed to be.
Absolutely.
But you don't know what, or do you?
No, we have some hypotheses. These gut microbes that we're talking about, they are known to live deep within the mucus. And so it might be that these specific microbes we're looking at have gone and just hidden within this mucus layer. And so they essentially have a protective shield.
And can you get at that?
Yes, so we have some ways to move forward. And one of our approaches that we can use is a technique called experimental evolution. And what this technique allows us to do is to look at the genetic changes that gut microbes acquire as they're routinely exposed to a stressor. So in this case, our linoleic acid is the stressor. When we do this experiment in a test tube, we see that gut microbes change the way that they metabolize fatty acids and they change their membranes. So then we can ask, are the gut microbes that have survived this exposure, do they have those same changes in fatty acid metabolism and in their membrane? And the answer is no, they don't. And so this again is highlighting that these gut microbes are not directly interacting with linoleic acid.
Sarah Durienzi, she works at Baylor College in Texas. Antibiotics undoubtedly save lives, but could we be saddling some individuals with a lifelong increased risk of a range of immune conditions if we treat them at a young age with antibiotic drugs? New York University's Martin Blaser thinks we are. Although they don't know precisely how yet, his team has found that perturbing the microbiomes of infant mice prone to developing autoimmune diabetes dramatically accelerates the trajectory of their disease, perhaps mirroring what we're now also seeing in human patients with type 1 diabetes.
This is a disease that has been accelerating in recent years. It's doubling in its incidence every 20 or 25 years. During the period of time when this disease has been doubling, antibiotics have come on the scene. And so antibiotics are a possible factor in this disease and other diseases like obesity and asthma. And the way they may be playing a role is by affecting the human microbiome, the organisms that live in and on the human body. We know that antibiotics perturb the microbiome. Our hypothesis is that perturbing it early in life is what's causing this big increase.
So what was the experimental method here?
We did our work in mice. There's a kind of mouse called the NOD mouse that spontaneously develops type 1 diabetes. And we wanted to see, could we accelerate the diabetes by giving the mice antibiotics early in life? And in fact, we found it could in male mice. There are differences between males and females, which we are exploring, but almost everything I'm going to tell you has to do with male mice.
Right, so you have cohorts of mice. You either give them, I'm presuming, a control where they don't get any antibiotics, or you give them a dose of, broad-spectrum antibiotics, and then you follow them up and you ask, do they or do they not have a higher or lower rate of diabetes subsequently?
Yes, you said it very nicely. We give the mice one course of a macrolide antibiotic very early in life, and then we follow them to see if they will develop diabetes. And here's where we saw that even one course of antibiotics both accelerated the diabetes, it happened earlier, and actually more mice became diabetic.
And can you show that in line with using that macrolide antibiotic, you also do genuinely perturb the microbiome and you perturb it in a consistent way in the treated animals?
Yeah, so our first findings had to do with the effect of the antibiotic on the microbiome, and we found very general kinds of differences that were consistent across mice. And we were able to name specific organisms that were significantly perturbed and that were associated with the development of diabetes.
Now, if that's the relationship that is causing the effect you're seeing, one would speculate then that if you put back the missing bugs, or you just recolonize these animals with the microbiome they had before you fiddled with the antibiotics, you should be able to defer the diabetes risk. Did you do that experiment?
We are doing that experiment now, which is to add back normal microbiota and ask the question, can we get them back to their baseline?
Type 1 diabetes being an autoimmune condition, if you're seeing an acceleration of the trajectory of the condition, you might think that it was some kind of loss of the normal gating process that's holding the immune system back. Do you think that's what's happening when you perturb the microbiome in this way?
Yes. So the next part of the study was to look at the effects of these changes in the microbiome on the host. We focused mostly on the intestine of the host, and our main focus there was on gene expression. Which genes are turned on, which genes are turned off. And our first observation was that when we look at mice as they get older, we can see a pathway of maturation. Over time, some genes are turned on and some genes are turned off. And what we found is that the antibiotic-perturbed microbiota very much altered this normal maturation.
There are a number of other immune-related conditions that appear to be increasing in incidence in the modern era, in line with things like diabetes and antibiotic use. Do you think we've got a common mechanism here then, that we're exposing infants to large doses of antibiotics at critical window periods in the microbiome maturation period, and that is having a knock-on effect for the way their immune system matures and therefore things like allergy risk and so on?
Yes, we agree with that too. We didn't study that in this paper, but it is consistent with the parallel increases in such diseases like asthma. And in fact, when we looked at the gene expression, we found many pathways that are seen in many different immunological conditions. We saw perturbation in these pathways. And we also found perturbation in metabolic pathways also, pathways about how the host is processing energy compounds, for example. So we think this is all tied together, both metabolism and immunity.
So are we medically robbing Peter to pay Paul? Because we're looking at the short term at how we keep young individuals safe and make sure that doctors don't get sued. But longer term, are we saddling that individual with immunological consequences because of our defensive practice?
Yes, so that's an extremely important question. First, let me just say that antibiotics are wonderful drugs. They're miraculous drugs. They've saved countless lives. But doctors and other health practitioners are using antibiotics more and more and more for very negligible illnesses in which it's thought that, well, since antibiotics have no real cost, no biological cost, we may as well try them. The mantra is, it may not help, but it won't hurt. But we're finding more and more evidence that it may hurt and that it may change the arc of development. That's very concerning because antibiotics are so widely used in childhood all over the world.
Is it too late then? If this has been done to an individual, is there any way of resetting the system, or has this ship sailed?
That's a very important question. We don't know the answer. That's why we're doing restoration experiments to see, can we restore? Can we get them back to their baseline? It's possible that the ship has sailed and that we have to focus on the next generation, on the kids who haven't been born yet, so that we can prevent this fate for them. We can prevent this problem from getting worse.
Sobering to think about, isn't it? Martin Blazer there. You're listening to the eLife podcast with me, Chris Smith. This episode, we're looking at the biology and pathology of the microbiome. Coming up, how your unique bacterial fingerprint gets established in the first place. But before that, according to the saying, you are what you eat. And to endorse that, it's looking increasingly likely that your microbiome might even affect how you behave in social situations. Mice uncolonised by bacteria, reared in so-called germ-free environments and born to mothers who are themselves microbe-free, tend to socialise a lot less than mice born normally. And in the brains of these microbially deficient mice, neurons in the amygdala, which coordinates fear and other emotional responses, express very different patterns of gene expression, suggesting that microbes in the intestines can influence how the brain handles information. Roman Stilling.
We were looking at the reasons that germ-free mice, so mice that are devoid of any microbiome, have social behavior alterations. They are socially impaired. What we find in a classical paradigm that is called the three-chamber social interaction test where you have one chamber with another mouse that the test mouse can interact with, or on the other side of the arena, there's another chamber that has just an object like a ball or an egg cup or something like that. And we know that we usually see when we just test normal mice that they interact a lot with their partner and less with the unsocial object. But with the germ-free animals, they interact less with their social partners.
So what was the additional set of questions you were able to ask of these mice when you did this?
In a previous study, we compared the gene expression in a specific brain region that we know is involved in social behavior to control mice. And we found that in the amygdala, this specific brain region, there were several genes differentially regulated that are associated with neuronal activity. Based on this finding, we wanted to see what is actually happening in a dynamic situation. So when they actually encounter a social interaction, what is then happening to this amygdala?
So how did you measure what the genes are doing in that part of the brain at the time that the animals are having their social relationship compared to when they're not?
What we need to take into account is that the gene expression is always a bit delayed. So we do this task just the normal way, put the mice into arena and let them explore. And then we wait 1 hour afterwards, take out the brain, and then we look specifically for the brain region we are interested in. We extract the new made RNA. Then we send this for RNA sequencing, which is a method to look at the whole transcriptome. So all RNAs that are in the cells that we analyzed at once with next generation sequencing technology.
So what do you find then?
In a normal mouse, when it experiences a novel situation, specific genes are switched on and off. What we find now in the germ-free animals is that we also see these pathways, and we also find something completely novel and unexpected, genes that are in themselves regulating gene expression by modifying the chemicals that are produced during gene expression, the RNAs. And these RNAs, when they are produced by the cell to make new proteins, then they need to be cut into the right message, right? And that's why they're called messenger RNAs. And this RNA processing pathway that was heavily upregulated in our study.
Interesting. So the way that the RNA is being handled in the cell is changing. How is that being affected?
We don't know exactly how this is happening, but we see that the genes that are coding for factors that do the cutting, these genes themselves are turned on and off to a much higher degree than in control animals.
And are these changes in gene expression profile and splicing profiles reflected in a structural or connectivity difference in the amygdala? So if you look at a sort of microscopic level, can you see any differences?
We have done this in another study and actually did find more synapses, a more complex dendricity in the germ-free animals, which somehow fits our narrative.
So how do you think that the message is transmitted between the microbiome, which is broadly in the intestine, and the developing nervous system?
To be honest, we don't really know, but I think what we need to focus on in the future is the immune system. We know that the immune cells of the brain, the microglia, they are also impaired in their function in germ-free animals. That, I think, is certainly a way that we need to look closer at to study the interaction of the bacteria in the gut with the function of the brain.
Now, if that doesn't redefine the phrase food for thought, I don't know what does. Roman Stilling talking to me from University College Cork. Vitamins are molecules that we can't make ourselves and that we rely on our diets to supply. But we're not alone, because many of the chemicals that we need, bacteria depend upon too, and this can sometimes bring us into conflict with our own microbes. Aaron Wexler's discovered how this works for vitamin B12.
We're fundamentally interested in answering the question, what does it take to be a successful gut bacterium? And so we know from previous work in our lab that we used a model gut bacterium, part of a genus called Bacteroides, that's almost universally present among human populations. One of the things it takes is the ability to take up the small molecule vitamin B12.
Now, when you say a good microbiome member, is that good for the bacterial outcome or is that good for the host?
So when I was saying a successful gut bacterium, I was speaking predominantly on the side of the bacterium. So they want to colonize the gut, and so if they are not doing their job right, they'll go extinct from that environment.
But equally, if they're too promiscuous and too greedy, there could be consequences for the host too, couldn't there?
That's certainly the case. Most of these bacteria are localized to the large intestine, and they seem to do just fine there. We get some benefits from them in terms of digestion, in terms of small molecules like vitamin K and folate. However, when they find themselves in the wrong location in the intestine, such as the small intestine, then we start seeing problems in people.
And do those problems include things related to B12?
Yes, so these bacteria are very good at taking up vitamin B12. One of the focuses of this paper was a particular protein called B2G that we identified, which is on the surface of these bacteria and has a very high affinity for vitamin B12. So when these bacteria with these proteins with high affinity for B12 find themselves in the small intestine and grow to large densities, then they start taking the B12 away from our proteins.
What, because that particular B12 binding protein is so good at doing its job, it's better than our own one. And so if you put the two head-to-head in a competition, the bacteria win.
Yeah, it's certainly the case in vitro. So when we take this human protein called intrinsic factor that we use to take up vitamin B12, give it vitamin B12, but then we incubate it with this bacterial protein, we find that in a very short period of time, the bacterial protein has taken almost all of the B12 away from this human protein. So we can reason that a similar process might be occurring in humans and in the small intestine when these bacteria grow to too large in numbers.
It's like throwing a bulldog and a poodle, a lamb chop, isn't it, and asking who's going to win. Why has the bacterium evolved to have this incredible scavenging power for B12 then? Obviously it's a valuable molecule, but this sounds like a case of overkill to die for.
That's right. I think it relates more to competition with the bacteria over B12 versus competition with humans for B12. So I think stealing B12 from intrinsic factor, the human protein, I think that's accidental. And I think it's a consequence of competition for vitamin B12 between gut bacteria versus between humans and gut bacteria. So these bacteria, like I said, they live predominantly in the large intestine, and the densities of bacteria there are very high. And so with high bacterial densities, you get a high level of interbacterial competition for nutrients. The human intrinsic factor has also evolved with humans over the course of our evolutionary history, and it's not used to competing with gut bacteria for vitamin B12. And so when these bacteria find themselves in high numbers in the small intestine where intrinsic factor is, it just so happens that these bacteria will win that competition pretty much every time.
So does this fill in that sort of clinical gap then? Because we knew that classes of patients do end up with a deficiency in B12 and they do have a bacterial overgrowth. And now this explains why these bacteria are able to render us B12 deficient.
I think it's reasonable to say that this is an explanation for that. We don't know for sure that this is happening in these patients because we haven't done the experiments to prove that. However, the in vitro data that we did collect suggests that this is a reasonable hypothesis.
Now, how did you actually stumble on this in the first place?
We were studying B12 uptake in these gut bacteria, and we found that a certain class of gut bacteria called Bacteroides contain the same four components involved in vitamin B12 uptake as E. coli does. And E. coli is the classic bacterium in which vitamin B12 uptake had been studied. So these Bacteroides species in our gut contain the same four proteins, but they contain extra genes that are adjacent to these genes encoding these transporters in Bacteroides. And these genes didn't have known functions. And one of these genes, which we called B2G, turned out to be this surface protein with high affinity for vitamin B12.
So what, did you do a little bit of conclusion jumping where you thought, well, they're in the same region, so they might be involved. So you then went and looked at them and found, oh, hey, presto, one of them really is involved in this, and this is the function.
Yeah, so we couldn't assume just based on their genetic location that they were involved in B12 transport. To get at that answer, we took a molecular genetic approach first by deleting B2G. And when we did that, we found that these bacteria struggled to grow in low concentrations of B12. The second clue came from studies we did where we found the intracellular concentration of B12 was lower in bacteria that lacked this protein.
So what are you going to look at next?
We have some clues that suggest that BTUG isn't the end of the story, that there may be other components at play, and we don't know what they're doing. Another thing would be to try to get at the clinical side, which is to try to figure out if BTUG really is the key piece that explains why small intestinal overgrowth of bacteria leads to vitamin B12 deficiency in people.
Aaron Wexler, he's based at Vanderbilt University. Now, what's more individual than a fingerprint? Here's Jens Walter to tell us.
One puzzling finding is that each individual has a unique and stable community of gut microbes that is almost as personal as a fingerprint. Studies have now tried to identify what causes this individuality and found that factors such as genetics, diet, environment, or lifestyle, or also, health or physiological state only account for around 30% of the variation. And even identical twins actually have different microbiomes. So this suggests that something unknown actually contributes to microbiome individuality. So what we tried in our study now is to determine the factors that contribute to microbiome assembly. And more specifically, we tested the hypothesis that differences in the order by which we acquire microbiomes early in life can actually cause variation later in life and the high level of inter-individual variation that we are observing.
Why do you think that the order in which we pick up microbes in this way might make a difference at all?
This is very much based on ecological theory that colonization history, or more specifically priority effects, actually influence the outcome of community assembly. Priority effects mean basically that an early colonizer will often have an advantage because it is occupying the niche first, and it will also have an impact on the ecosystem by influencing colonizers that come later. Due to this, the order or the timing by which these members in a community arrive will then shape how the community looks in the end and how it functions.
I suppose it's a bit like if you walked into a room and there was a bunch of empty seats and there was no one sitting in any of them, you'd choose any seat to sit in. But if you walked into the same room and there were 2 or 3 people sitting down already, you might change where you sat and who you spoke to.
That's an excellent metaphor actually for what we are feeling. Yeah, and in ecology, we are actually referring to this as niche preemption. The one who comes first basically sits first.
Now, how did you test this, though? Because it's a nice theory, it's a nice idea, but how can you subject that to proper scientific scrutiny to find out if that's really what's going on?
So we have given this quite a bit of thought. So what we did is we used mice that don't contain any microbes, meaning they were germ-free. And these mice were further genetically identical, and they were housed exactly under the same condition using airtight plastic bubbles. So what we were basically trying to do is we controlled all of the experimental variable that we could think of. We then associated these mice with either entire microbial communities or specific bacterial strain cocktails in combination with communities, and we introduced these entities in different succession. We could specifically test if the timing and order of early life colonization influences how the bacterial community is composed.
What about the immune system, though? Because I put it to you that when I put a group of bacteria in, they might do something to the immune system, which that in turn affects the receptivity of the location for whoever comes in next.
Now, that is an excellent question, and we have actually given this quite a bit of thought. We think that early colonizers can somehow interact with the immune system and actually induce tolerogenic immune responses that would then favor this early colonization. To test for this, we actually repeated our experiments in mice that do not have an immune system and therefore, cannot actually at least develop immunological tolerance to these early colonizers. And when we did these experiments in these immunodeficient mice, we still found a clear evidence for priority effects.
So what are the implications of this then? Because We've got lots of babies popping out all around the world every day. Not all of them are being born via the normal route. Some are being born by, say, caesarean section. Some are being given big doses of antibiotics when they're born, usually for very good reason. But that could therefore have, based on what you're finding, very profound effects on the future microbiome structure for those individuals.
We can make two major conclusions here. First, as you said, we get an understanding about what actually shapes our microbiome. And since we showed that the early acquisition is important, clinical practices that influence this early acquisition of the microbiome, such as caesarean sections or antibiotics, formula feeding, will actually then influence how the microbiome look later in life. So appreciation for these ecological events early in life might help us to develop different clinical practices to potentially prevent aberrant assemblies of gut microbiomes. The other main thing that we learned is that if we want to modulate the gut microbiome more permanently, we probably have to do this early in life, because early colonizers have a higher success of actually establishing themselves in the ecosystem. And not only this, these early colonization effects might also allow us to specifically alter the entire community, establish more healthy microbiomes, or prevent the establishment of microbiomes that might have a detrimental effect in the end.
Jens Walter from the University of Alberta. And there we must leave it for this month. You've been listening to the 50th episode of the eLife podcast, all 49 previous editions of which, together with full text transcripts and details of how to subscribe, are available on the Naked Scientist website, which you can find at nakedscientist.com/eLife. I'll be back meanwhile with another look inside eLife next month, including how the Tasmanian devil facial tumour disease has reinvented itself. Until then, thank you for listening, and from me, Chris Smith, goodbye.
The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.